Good afternoon, viewers at home. My name is myself. I am the CEO of Bio-Edutainment. This afternoon, we are going to continue our educational series discussion with a center. Topic of discussion is going to be on the carbohydrate system of classification. I told you that in the earlier video that carbohydrate made an attempt to classify all organisms to belong to three domains of life. So you are quite aware of this before now. So now the same roots, all these three domains, they have the same roots. So and the first one is Eukarya, which is the largest in terms of multicellularity. We are going to see it here, followed by Prokarya according to their cellular makeup. Prokarya, or what we call Prokarya, and this one is the smallest Eukarya. Now, according to me, it makes use of some small subunits ribosomal RNA. He made use of the sequences, and he collected all the sequences of all organisms, and he was able to do the molecular analysis of it. And when he did the molecular analysis, he was now able to classify them based on these attributes. So don't forget that in the earlier video we actually talked about classification of organisms. So and here we made mention of the system, the traditional system of classification, that is the classical way. So after that, that was when Carl came up with the use of the phylogenetic tree in the classification. And the earlier video we had maybe sometime yesterday, so I told us about phylogenetic tree, what they are, the types of phylogenetic trees include rooted, unrooted, and so on. Then apart from that, we now look at what are the basic applications of phylogenetic tree well as medical clinical microbiology. So today, just like I said, this is another good application of biology. So according to we divided them into three, and let's now look at each of these one after the other. In the other video after this one, we will start speaking them one after the other. But our center of discussion is going to be majorly in this class, and in this domain of the Prokarya, because we only have few members of care that are microorganisms. All of them, most of them, let me say 70% or 90% of them of the Eukarya, they are macroscopic, that is, we can see them. So that is why our center of discussion is going to be on Prokarya. And don't forget, because this book is microbial information. So that is why our center of discussion is going to be based on that. And what we will do immediately after considering each of this one, we will look at what are the organisms Prokarya that fall into low G plus C, and we look at this one that fall into high G plus C. After we just make mention of this as a passing statement of representative of Archaea, representative of Eukarya, we just make mention of them so that in case we are asked to compare them and give them basically distinction. Now, in today's lecture, we only consider three features of each of these class. We are going to five, minimum of five. So we look at B, we look at E and N. Under B, we look at what are their medical importance. We highlight them. Then we look at what are the members. So we look at giving them members. Then here we will now look at how, if you have any infection, how can they be diagnosed and treated. So diagnosis and treatment of each of these. We look at Eukarya, that is with 1 A, B, C, D, E, F. So we also put the second domain. We look at A, B, C, D, E, F. So then we also look at Archaea. We put A, B, C, D, E, F. So before we will now go into considering them, our center of discussion will now be on the domain Prokarya, and we are going to look at low G plus C as well as high G plus C. So the first thing we are going to know about the fourth domain, structurally, we have very complex internal structures. Very complex. The structure is very complex. The level that by the time we go to the cell, by the time we go to the cell, we see that we can do the neurotrophs. similarity. That is according to the theory of Eukaryotes. So they have swallowed a lot of cells, now making them to contain. Don't forget that all these organelles evolutionarily were small cells that have existed ever before. So as a result of the symbiotic relationship, that was why the bigger one, which now form the plasma membrane now, swallowed the other smaller one. And the other smaller one, in an attempt to exist as independent entities, they have become organelles that are capable of performing their independent function as well. So you need to know that. Then another thing is that this is the only domain that we have sexual reproduction being pronounced, highly pronounced growth. So the cellular element, so because their cell that is the non-distinct into male and female damage. So sexual reproduction, they reproduce sexually with them. That the majority of them reproduce sexually. Although in smaller ones like yeast, we still have other asexual form of reproduction like breeding. But majorly sexual reproduction is being pronounced here. Then the next one is that the genetic material. The genetic material is highly located linearly inside the nucleus. So because nucleus itself is another so that is why when you know yourself, you may discover that inside the nucleus, that is where we have genetic material, which is DNA, is mainly located. Then, just like I said before, in terms of complexity, they are very, very complex. They are complex because the highest level of organization is found in Eukarya. So they organize themselves into many cells, and these many cells form tissues, form organs, and these organs also form system. Before combination of the system will now form the whole organism, just like what we see. So in terms of complexity, they are highly complex. Just like I said here, just like their internal structure, even in terms of cellular organization, it is in this domain that they are highly complex. Then the next thing, which is in terms of their evolutionary features, in terms of evolutionary distinction, we notice that the first thing I mentioned, I've even mentioned is that when you look at the structure of chloroplasts and you look at the structure of mitochondria, you discover that these chloroplasts and mitochondria, they are from the same parents. They are from the same parent, and when you now compare them structurally, they resemble the prokaryotic because when you draw, then you begin to be like this. This is the prokaryotic organism that is bacteria, and when you also draw your mitochondria, you draw mitochondria almost in the same shape, as you have there in prokaryotic organism. So because, and one of the reasons is because within prokaryotic organism, they have their own DNA. They have their own chromosomal DNA as well. Although their DNA is not actually located inside the membrane boundary difference, but they have their own. So similarly, the chloroplasts as well as the mitochondria also have their own DNA. You can see a kind of distinction, a kind of similarities between them. So evolutionarily, we believe that Eukarya arising from prokaryotic counterparts, the prokaryotic counterpart, because they still retain prokaryotic counterpart, they still retain their prokaryotic attributes that forms chloroplast in plants as well as mitochondria in animal cell. So and this eukarya is actually differentiated into 4 different groups. Like that they belong to they have 4 different kingdom. So the first kingdom is the kingdom animalia, followed by the kingdom plantae, followed by the kingdom fungi and kingdom protista. So we only consider these two in this course, the fungi, the phylum Microbial, so the fungi, and some as the passive statement we can also come across those ones that are representative of protista. So just like I said, the other lectures we will continue having the kind of comparison. So we look at the B, E and F. Now let's now move to the domain prokarya. Domain prokarya, just like what we did for eukarya. Structurally, prokarya are made up of cell wall instead of cell membrane. They are made up of cell wall, and inside their own cell wall is a compound that is called peptidoglycan. Peptidoglycan, when we look at the cell wall, and this is the presence of peptidoglycan, either thick or thin, is actually what we need them to biochemically to differentiate them into gram positive or gram negative, and in some cases they may not take up gram. We use different dyes for them because they contain structurally they are made up of cell wall that contains peptidoglycan, peptido for peptide, and glycan which is carbohydrate. We are going to the structure of all these that contains NAM and NAD. So NAM, NAD, okay, so that's not part of discussion today. Then number 2 is that they have no membrane bounded organelles. Their own cell are not differentiated into small organelles. So they don't have membrane bounded organelles, which is completely absent in the main prokarya. So then another thing is that unlike what you have in eukarya, this prokarya or prokaryotic cell contain single cellular DNA. So single cellular DNA, which is located in the area called nucleosomes, but not nucleus as such. So nucleus is an area, a kind of area that can be regarded as a nucleus, but they are not membrane bounded, and their DNA is single and circular. Unlike although in some prokaryotic organisms like E. coli and some others, they have what is called extra chromosomal materials that they actually use for survival, especially for antibiotic resistance and some other things. So you should know that. Then in terms of their size, they are smaller. They have smaller sizes compared to their eukaryotic counterpart. So in terms of their distinctiveness, in characteristics or uniqueness in their characteristics, this is their own life is a life of unicellularity, is a life of independence, is a life whereby a single cell can persist throughout their lifespan. So everything that eukarya will do by cooperation between the cell, you see prokarya being very simple, but they make use of just one cell to be able to perform all the function, almost all the function that is going to take many cells or eukarya or eukaryotic organism to perform. So that is it depends on the life of the unicellularity. Although, okay, so then here asexual reproduction is highly pronounced. Asexual reproduction is highly pronounced in the prokaryotic cell. Until maybe some years back, that was when they now discover some genes, or let me say some appendages, form of that we can that we call F-pilus and F-negative pilus and F-this that can be regarded as their own values. But predominantly, members of the prokaryotic prokaryote is circular, not linear. You have it linear here, but here it is circular. And metabolically, they are more competent in terms of their diversity. Metabolically, they are more competent in diversity than just one eukaryote. So that is what you should know. So we have different pathways that they use for different purposes. So then evolutionarily, they are the oldest male. They are the oldest form of life, and it conjugated, their existence conjugated to around 3.5 billion years ago. So it is the domain Eukarya that are the earliest that will to come. Okay, well, because they are related, not the earliest, they are related. So this one existed, must have existed around 3.5 billion years ago. That is, if you are comparing them with Eukarya, they are what? They are older than Eukarya. And in terms of their ancestor, they are the ancestor of the eukaryotic organism. They are the ancestor, because Eukarya must have, just like the theory of endosynbiosis, just like what I said here, they are the ancestor of the eukaryotic organism. So also this one will also continue to D, E, and F. So and that is going to be the second domain we are going to consider. The third domain which happens to be the least in terms of population, the least in terms of population and representative as well. Structurally, they are similar to prokaryotic organisms. In fact, it's almost the same way you see prokaryotic organism as non-membrane bounded state. They are closer in terms of structure to prokaryotic counterpart. Then in terms of membrane bounded organisms too, they don't have membrane bounded organisms, they don't have any distinct organism, they don't have anything that will actually be regarded as organelles. Then their cell wall, instead of it to be made of ectoglycan, it contains a different compound that is called pseudo-peptidoglycan, or it can be entirely made of protein. So don't forget that part of the organism of this one are prions and fluids. So we will see all this, we will mention all this maybe later on, or you might not even mention them, because just like I said, our center of discussion is around the main Prokarya. So then in terms of sizes, their size are small as what we have in the Prokarya, and even smaller sizes than prokaryotic organisms. Okay, so then in terms of their distinctive features, they exist instead of existing at either unicellular level or unicellular level. So this one exists as the extremophiles. Everybody knows that they exist in a very harsh environment, very harsh and high temperature environment. Another thing is that they are unique biochemically. So most of the chemicals that even prokaryotic organisms will utilize for their metabolic diversity products, we already have them in Archaea. So they are unique in terms of biochemistry. So then most of them they are mechanogens, so they know how they can produce mechanogenesis. So it is in Archaea that we find them. So because of space, we ought to have talk about Archaea at evolutionary feature. What we believe is that Archaea is the simplest. So Prokarya also originates from Archaea. So just like this, evolutionarily, Eukarya came from Prokarya, and the Prokarya also comes from evolutionary. So we should know that. So the most primitive, because even at this level, this one can also exist as particles. Sometimes we think that they are not living, but they exist as particles. That is the lowest form of life. So they come from their prokaryotic they come from Prokarya, come from Archaea, then Prokarya will now give back to this in terms of their evolutionary relatedness. In fact, at this point in time, at this level, that is where they regard RNA as an entity. That was when they started the theory of RNA world. That is, RNA can also exist as independent before the issue of the DNA was even being discussed. So some of them, so we have seen at Archaea level that some of them exist at RNA level. We see some of them that exist at the DNA RNA level, and we see some of them that exist as only proteins. So at this level, that is the lowest point by which life can actually exist. So just like I said earlier on, today's topic is actually today's lecture is actually focused for 3 domains of life, and we have highlighted them according to karyotes, and what we use for the small subunits like the human RNA, the sequences. That was what we use to classify them into Eukarya, Archaea, and Prokarya, and we have the common which is their ancestors, and we have examined what are the distinctive characteristics and what should we actually know about Eukarya. We have highlighted them, and out of it all, we still see that the most complex out of the 3 Eukarya, followed by Prokarya, and the simplest is that of Eukarya. So, and just like I said, in terms of comparing the two, which one has existed at highest level of organization, which one exists. So here you have them existing at cellular level. So you don't have anyone that exists at tissue level. So that is why cellularity is the main achievement of this. Then we have to look at that one. So in the other video, we are going to also compare them in terms of what are the medical importance of each of these. Medically, how can we diagnose them, and if there are any infection, how can we be treated? Then we will consider also basic examples of this one. So don't forget to comment in the comment section, like this video, get notification, as well as subscribe to my YouTube channel. There are still other educational videos that will still be coming for. So any video for now, I'm going to meet you at the feedback level, where I'm going to send you a lot of prayers and having